Hi, I'm Maiko Uemura from CNDR. Today, I'd like to talk about the distinct characteristics of late in Lewy body disease. As you know, TDP-43 pathology as observed in some older adults calls out late. And the late frequently coexists with other neurodegenerative diseases, including Alzheimer's disease and Lewy body disease, and both are reported to be associated with cognitive decline. But it is unknown the morphological and distributional differences between late LBD and late AD. So we try to see the characteristics of late LBD by comparing it with late AD. First, we studied the morphological differences of late between LBD and AD in the hippocampus. We found a different distribution of TDP-43 neurocytoplasmic inclusions, or NCI. While LBD had more NCI in CA3, AD had more NCI in the CA1 to ceviculum. And we also found that some late LBD cases has abundant TDP-43 fine neuroids in the CA2 ceviculum, while the number of NCI in the CA1 was higher in late AD. The fine neuroids in the CA1 was more abundant in the late LBD. These are the double immunofluorescence of TDP-43 in green and alpha-synuclein in red. The TDP-43 fine neuroids were most abundant in the CA1, but CA2 was the most frequent region having TDP-43 and alpha-synuclein co-pathology. Next, we studied spreading pattern of TDP-43 pathology, examining 16 regions and ordering them according to the frequency. To stage it, we applied conditional probability analysis. The result shows there can be 4 stages for late LBD. First, the amygdala at stage 1, followed by the hippocampal region at stage 2, the brainstem and the cingulate at stage 3, and finally the subcortical and the neocortical regions at stage 4. By applying it to both LBD and AD, we found 2 major differences. First is the dentate gyrus. In late AD, the frequency of TDP-43 in the dentate gyrus was less than other hippocampal regions, and the dentate gyrus was separated as stage 3. But in late LBD, the frequency in the dentate gyrus was almost the same as at hippocampal regions and included in the same stage as stage 2. Second is the brainstem. While the frequency in the brainstem was less than temporal gyrus in the late AD, it was more observed in the late LBD and was placed in the earlier stage as stage 3. Finally, we examined the cognitive decline in the late cases in each cohort and found that cognitive decline is accelerated by late. We also studied the last MMSE in LBD with breaking down by LBD subtype and late stage. The MMSE score got lower according to the LBD subtype, but late partial cases showed lower MMSE than late negative cases in each LBD subtype. And the MMSE score got lower according to the late LBD stage, suggesting that the occurrence and the prevalence of late affect cognitive impairment in Lewy body disease. This is the conclusion of this study. Late LBD and late AD are morphologically and distributionally different, and the LBD subtype and the late LBD stage are associated with cognitive impairment. This study was done with a huge help from many people. First, I'd like to thank Dr. John Trojanowski and Dr. Virginia Lee and other CNDR members. And I'd like to give a special thanks to Dr. Edward Lee for directing this project. Thank you.